When did you learn how to walk or read? You will probably say when you were a child. A lot of the skills that you have and so effortlessly use today can be credited to a time during your childhood called the critical period, when your brain had unlimited potential. When we are born, the different connections in our brain are like puzzle pieces, full of potential ready to be arranged. This is when the critical period begins, when our brain becomes really sensitive to the environment around us. And the experiences we have during this time help to arrange those puzzle pieces or brain connections into a structure that can process and remember those experiences. As we reach adulthood, the critical period closes, and the brain connections we form during this time begin to stabilize, giving us the capabilities we will need for the rest of our lives. The benefit of the critical period is that it helps us to develop a brain that is perfectly adapted to the environment we live in. But the downside is that any negative environmental influence during this time can lead to an unhealthy brain organization, which can contribute to neurodevelopmental disorders like autism and visual disorders like amblyopia or lazy eye. Now, let me tell you how we study this in the lab. Previous studies have shown that if mice are raised in the dark and go through the whole of their critical period without ever experiencing vision, then their brain's ability to process visual information is permanently altered. However, if you're able to introduce these mice to light during the critical period, even for a few hours, then their brain is able to respond to that experience, build the connections that are necessary for vision, and the animal will have a complete recovery of their visual function. This is the power of critical period. So, the million-dollar question is, can we reopen this critical period in adulthood so that the brain of an adult mouse can also respond to new experiences as well as a young brain would? This is the question I'm trying to answer in my PhD. I will be studying the changes in the brain of mice that are raised in the dark and see if giving different treatments in adulthood can reopen critical period to promote recovery of their visual function. To study this, I will be using a very exciting technique called the two-photon microscopy, which is a specialized microscope that will allow me to look inside the living brain and study detailed structural changes in individual cells and their connections before and after giving the treatment in the same animal. So, my study is all about turning the clock back to a time when the brain had unlimited potential. With this idea in mind, I hope to identify potential treatments for neurodevelopmental disorders that will give the brain a second chance to build healthy connections.